Good day to everyone, and thank you for registering to this live webinar. We're hosting this webinar with attendees from all around the globe. Welcome, everyone. My name is Ricardo Sauer from University College London, and I will be hosting this webinar on behalf of the Early Career Members Network of the Metabolomics Society. This is actually the 16th of our webinar series session, and it's brought to you by the Early Career Members Network Committee. Currently, the webinars are free for all to attend by the courtesy of the Metabolomics Society and our invited speakers. And I would like to inform you that all the material will be recorded and uploaded to the Metabolomics Society website, allowing the access to the live content at your own convenience. Please connect with the Metabolomics Society on Facebook, Twitter, and LinkedIn, as you can see on the screen. I would also like to ask you for feedback about the Early Career Network. You can share your ideas and suggestions at the email address info.emn@metabolomicsociety.org. Before introducing our speaker, I would like to introduce you to the new Metabolomics Forum. It's a platform where to talk and discuss any subject related to metabolomics, either Metabolomics Society, hardware, software, databases, and so on. If you have any questions related to the metabolomics, you can post it on the forum and get answers from the users who are experts in the field. Please feel free to visit the forum, create your own profile, and use this platform to talk about metabolomics. The link is displayed on the screen. Finally, I would like to introduce as well Metabolomics 2018. The annual international conference of the Metabolomics Society is coming up in June from the 24th to 28th in Seattle, USA. The network will be actively present at the conference. Some workshops are currently being organized by the committee. Well, I would like to introduce our speaker. Today's expert speaker is Professor Sauer, who is a professor in systems biology and the head of the Institute of Molecular Systems Biology at ETH Zurich in Switzerland. The research of Professor Uwe is focused on the coordination of metabolism, bacteria, and yeast, as well as metabolic feedback in the regulation systems. The Sauer lab actually has pioneered on the development of quantitative mass spectroscopy-based methods for metabolomics and flux analysis. Computational methods have also been used to generate some hypotheses on biochemical mechanisms and metabolic coordination. Well, there will be time after the webinar for questions, so please send your questions by using the questions option on your web app. You can type the questions throughout the presentation, and our speaker will aim to answer as many as possible at the end of this webinar. I think we're ready to start, and I will pass you on to our webinar speaker, Professor Uwe Sauer.
Okay, good. So welcome, everybody. I thought I would focus on an aspect that in metabolomics we don't normally focus that much on, and that's trying to make sense of the data. A lot of the metabolomics meetings where I go to, it's much about the techniques and being able to measure more. And that's a very natural development in basically any omics method, including proteomics and transcriptomics. But I think we are already at a level that we can measure a lot. And it is time that we spend a bit more time on being clever about generating hypotheses. And that's really what I want to focus on today. And in particular, hypotheses and trying to understand how is cellular metabolism actually regulated. How do the cells make decisions? And I will focus only on two aspects. In the first part, I will talk about steady state analyses and use a method called correlation to look at, to generate hypotheses on potentially regulating metabolites. And in the second part, it will become a slightly more mechanistic when I focus on dynamic data and we use a simple ordinary differential equation. Okay? So to get started, metabolism, as probably most of you are aware of, is really probably the best understood and known, maybe known biological network to date. After decades of discovery, we have a huge amount of accumulated detailed knowledge. And in the recent years, it became more and more clear that metabolism is not only a dull workhorse that is being controlled by regulators, but also actually sending out signals to the regulators and therefore informing the cell about its current state. And so this is where we start. So we know very much about what you see here in the center, the stoichiometry and the topology of these networks. You all know that one can generate genome scale networks of metabolism. And what we are interested in my lab is which of these known up here molecular interactions between proteins and DNA or metabolites and proteins is actually making a decision for metabolism so that the cell switches from one scenario, maybe directing the flux as you see it here, to a situation like this. And so we're very good in the stoichiometry and the topology, but we're actually very poor in understanding what would happen if you add a drug, or if you change one regulation event, or even know which regulation event does what. So that understanding the dynamics and being able to predict metabolite changes is one of the weaknesses of the metabolic research field in general. So in my lab, we focus on metabolite protein interactions. And the problem is the metabolites are input to proteins that can be enzymes or regulators, and we'll see both examples. But at the same time, these proteins change metabolite levels and fluxes, and therefore there is a feedback loop, and that's very difficult to sort out. And let's start with something simple. What is our present knowledge of metabolite enzyme regulation? So only metabolites that are acting as allosteric regulators. So we recently put together a framework that allows to basically reconstruct from the biochemical literature and from databases such a regulation network between metabolite and protein. So if you're interested, down here is the reference if you want to read this up. All I want to say is that we now have a genome scale architecture of E. coli small molecule regulation network. And it's about 1,700 unique regulatory interactions. About 300 metabolites affect 350 enzymes. Most of them are inhibitory, and just about every other enzyme is regulated. Okay, so it's already, that is a lot. We know already this is only the tip of the iceberg. There's much more regulation coming. But already that is a solid basis of knowledge. And so the result of this kind of knowledge is what you see here. It's only a part of this large network. I'm focusing here on the E. coli central metabolism. You will see glycolysis here, the Krebs cycle, TCA cycle. And all you should be seeing from here is that in the central part of metabolism, essentially every enzyme is activated or inhibited by at least one compound, typically by many. So that's nice. Now we have a topology. We see, we have a list basically and a network that shows us how the interactions are. And we can start to ask questions that really biologists care about. So for example, if you want to redirect the flux and we want to change the regulation, like the analogy to if you want to redirect traffic in a city and change the traffic lights, which ones do we have to change? And what do we have to do exactly? And this is where basically we don't know anymore exactly what we have to do. And this is not only so for metabolite protein interactions, but in general, we know from the literature that there's literally thousands of regulatory interactions that were identified in individual experiments, but we understand very little which of them matters to actually make a specific decision. And that's where the starting point for the first story is. And we asked the question, how does E. coli coordinate its transcriptional regulation network? And so this is work that was done by 3 PhD students in the lab, Carl, Luca, and Dimitris, who took up this question. And you probably all know there's a transcription factor network that's relatively well characterized. So it's about 200 transcription or 300 transcription factors that regulate the expression of several thousands of genes. And we know the transcription factors, some of them listen to external signals, but many listen to or are responding to intracellular metabolite levels. So there's two types of input, and many of these transcription factors here, they regulate enzymes in the metabolic network. And so the questions these 3 guys were asking is, which metabolites are the signals? How are these signals now integrated to lead to a sort of a coordinated response that makes sense to the cell? And while I'm talking, you can think yourself and take a guess. If we want to look at 30 different conditions, how many such metabolite signals do you think are necessary to regulate the central metabolism of E. coli? Take a guess, write it down somewhere, and then we'll see what the answer is, and we can compare what you found. So before we can go into this, I have to take one step back, and that is to say that gene expression depends not only on the transcription factors, this is what I will call specific regulation, but also on what we call global cellular regulation. And that is, if you have a gene, the transcription factor may bind or not, but also there needs to be enough RNA polymerase. And that RNA polymerase is not always available at the same frequency. And so it depends very much on how fast the cells are growing. And so about 4 or 5 years ago, there were 3 papers that quantified this aspect, and that all 3 agreed that most of the transcript changes that we measure in a cell are actually driven by the availability of the RNA polymerase. And that is directly correlated with the rate of growth. And just to give you an impression of how much a gene expression changes, even without transcriptional regulation, here's 12 synthetic promoters that were made so that they have no binding site for any known transcription factor. So they are what we would say constitutive. And each dot here is one expression level measurement by GFP. And down here is the growth rate. And this is the activity that we measure on the y-axis. And you can see if they were constitutive, you would normally expect it's a straight line. There's no change in activity over growth rate, but you can see all of them show hyperbolic curves and change by some of them an order of magnitude, but at least several fold the expression level, even without transcription factors. So to do something in trying to learn what metabolites do for transcriptional regulation, we have to take care of this global effect. And so we do this in the following way. So the experimental setup that I'm going to discuss now is we only focus on central metabolism. So we picked 100 GFP-based promoter activity probes, essentially covering all enzymes of central metabolism. We use targeted LC-MS Metabolomics. This is a standard method we've been using for a while. It's very targeted to central metabolism. We can generate absolute concentrations for about 100 metabolites in central metabolism. And we do this under 26 steady state conditions. And we use now a mathematical formalism that allows us to remove the global regulation from the specific one. And that is in log space, the logarithmic change in promoter activity is the sum of the logarithmic change of global and specific transcriptional regulation. Okay, so this is what we measure. If we now find a way to know what is the global, we know what is the specific effect. And so we do this by a simple hypothesis for if we compare all promoters against each other. If a promoter was only responding to the growth rate, we would expect both of these promoters to fall, the expression level, their expression level to fall on the diagonal. Any deviation from the diagonal between these 2 promoters would be an indication, at least, that this is specific regulation. Okay. So then we use linear decomposition as, by singular value decomposition to basically find out these specific regulation events here. And so I don't want to bother you with the details of this heat map, but just so that you know what it is, what we have 26 conditions that they are listed here on the left side. Many of them are different carbon sources where the cells grow at different speed. Then we add also chloramphenicol because that by this on the same medium we block the rate of growth slowly. We don't block it, so we slow it down. So it's a sublethal inhibition of the ribosomes. And then we measured the GFP-based promoter activity over all the different parts of central metabolism. And so each dot in one of, I'm showing you here one of these, for one of these promoters for the GLPA, the activity probe, growth rate as a function of the normalized promoter activity. And you can see under most conditions, this particular promoter is actually not active. There's only one when it's really active, and that makes a lot of sense. This is the transporter for the carbon source that was added. So this is the simplest case. Off most of the time, only on when it is needed. And on the other extreme, I'll come back to this example in a second, we have a case. We know this is constitutive because this is an included constitutive promoter, a synthetic one. it really does not, it changes its expression level a lot, but it is linearly correlated with the rate of growth, okay? And then there are some other patterns, and we're interested afterwards in the blue dots that are really deviate, or any of the dots that deviate from these straight lines. Okay, so now we have a measure of the effect on gene expression that's global. And if you look at all the 100 genes roughly that we have, about 70% of the gene expression changes that we see is already explained by simply changes in the rate of growth. And this is illustrated here. So here we have the promoter I just showed you, a constitutive promoter was a very good linear correlation across many different conditions. Doesn't matter if we inhibit the red dots, the ribosomes, with chloramphenicol, or if it's different carbon sources. The expression is really only a function of the growth rate. So the promoters in this range here are primarily, or in part even exclusively, regulated by global regulation. And down here now, we have those that are for us interesting cases, because here we see strong deviation from the diagonal, and here we would now expect there's a transcription factor that is responsible for the specific regulation of this gene. Good. Now we can come back to what we actually wanted. So far we have, from our measured data, global and specific regulation, we removed now the portion that is global, so that we end up now with only the specific regulation for each of the 100 promoters. Okay, so this is now a cleaned up version, and we're going to work only with this. And now we ask the question, without knowing anything about transcription factors, is any of the metabolites that we know correlated with this change of specific transcription factor regulation? And so if it was correlated, we could take this as a first hypothesis that this metabolite is actually activating or inactivating the transcription factor that is responsible for starting expression from this particular promoter. Okay, so because this is now a simple test, it's a correlation. We can do this systematically for all metabolite promoter pairs. All of the 100 GFP promoters with about, I think, 70 metabolites are measured in this case. And in this particular case, for pyruvate kinase F, we see a very good correlation between one metabolite and the expression of this promoter. So I'm cutting this now very short and just show you the results, because that has just been published last year, if you're really interested in it. But basically for every, up here is again the transcription of the different promoters grouped into glycolysis, gluconeogenesis, TCA cycle, for example. And here are some metabolites, only the top 12. In fact, there would be now, if you go down here, there would be about 70 different metabolites. And mostly you see it's white. That means no correlation of any metabolite with the clean expression, with the specific expression of this particular promoter. But there are some where we find a very good correlation. And so the good correlation is sometimes positive when it's activating. So the green ones are when there's a positive correlation, when the more of a metabolite, the more expression is there. And the red ones are would be repressive, the opposite effect. And everything that has these solid boxes were actually things that were known already. And the open box without the solid line are the ones that are now new discoveries. So the summary of this is we find only very few metabolites that actually correlate with promoter activity. And we capture, that's very good for us, we capture many of those that are known. For example, these green ones here, the green ones up there. But we also discover novel ones. And in principle, this is applicable to any organism. So far, we didn't need to know anything about any transcription factor. But now we do, because I want to show you something else. So what you find here is 3 metabolites really explain most of the specific transcriptional regulation. So it's cyclic AMP. You see there's many promoters that respond either positively or negatively to the levels of cyclic AMP. And then there is about 11 that respond to either fructose 1-phosphate or fructose bisphosphate. And these are not just random. So I should say down here, the other metabolites is almost exclusively white. So I show you only the part where something interesting is actually happening. So these metabolites are not by chance, and because we work with E. coli, we can now go back and say, and ask, oh, which metabolite, which transcription factor is known to respond to these metabolites? And then cyclic AMP is actually, there's a cyclic AMP receptor protein, a well-known, very well characterized protein that is a transcription factor that is known to respond to cyclic AMP levels. And you see that we find that activation, but we also find novel interactions of cyclic AMP, in fact, in the opposite direction of what was known so far. And the two ones up here are the input to the transcription factor K. Both are known, very well known regulators of central metabolism. So what we have learned is that from actually relatively simple correlations between GFP expression or could also be mRNA expression levels by other means, and metabolite concentrations, we discovered about 30 novel interactions between transcription factors and metabolites. The benefit for E. coli is that most of these interactions are already known, and so what we really want to understand is, yes, it's known, someone wrote this in an article or in a textbook, but what does it really do? And so this textbook knowledge that does not help us to understand what actually matters under what condition. And so what we find now is that, remember the question that I asked you, and now you can look at how many metabolites you need to coordinate E. coli central metabolism. So at least central metabolism under these 26 conditions, more than 90% of the gene expression is explained by global transcriptional regulation through the growth rate, and two transcription factors, CRA and CRP, and they're coordinated through only 3 metabolites. And that is enough to explain the vast majority of the transcription changes. Okay, so I don't know what you guys thought, how many we need, but at least this is the answer that we find for central metabolism. And for me, the conclusion is at a higher level that E. coli does not micromanagement the expression of its central metabolic proteins. It takes very simple input, and that provides it with a generally right amount of all the different proteins. Not perfect, but generally okay. And that means also that there is simplicity in the apparent complexity. So what I showed you before was huge amount of regulatory interactions, but we don't need to understand all of this in detail. In fact, if you want to re-engineer the cells, we should really focus on those 3 metabolites and those 2 transcription factors. Okay, so that's the first part, and I would like to move a little bit into a more mechanistic direction, but still looking at metabolic regulation. But now we're back to the slide that I showed you before, E. coli central metabolism, enzymes, and many of these enzymes are regulated by many different metabolites. So which of these that we see here on the picture is actually now important? When does the cell need it? And what exactly does it do? And how strong? Are many of them needed, all of them, only one or a few, to explain the dynamics of metabolite levels. And that's what we in metabolomics measure. We measure the concentrations, but eventually we want to understand why are they changing the way they do, and what are the changes then telling the cell. So to get from this kind of a static representation to understand the in vivo function, in my lab we use heavily metabolomics, but also modeling, and we need the modeling. And one point that I really strongly want to advocate is steady state measurements for metabolomics is a very difficult to interpret quality of data. We've done initially a lot of steady state experiments, but the levels of metabolite changes are not only the consequence of a particular perturbation, they are also now afterwards affected by a lot of secondary consequences. And so it's a little bit like with gene expression. If the rate of growth changes, not only the gene expression changes, also metabolite levels are changing. So what we learned is that dynamic data is extremely important, and that's what we're going to focus on now. Because it's important because it allows you to delineate direct from indirect effects. So Hannes and postdoc in the lab and Karl started to address the question, can we infer active allosteric regulators of enzymes from the effects that they show on the metabolome? And so allosteric regulation, just to remind you, if we have an active site here, if now a metabolite binds at some other site of that protein, it changes the status of the active site and therefore makes the protein less or more active. And the assumption that underlies the work that I'm going to show you is that such binding effects, allosteric binding effects, they would be really important for very rapid responses. And that's what I'm going to show you now. So we used a very special design. We borrowed the idea from Josh Rabinowitz's lab in Princeton. So we cultivate the cells in a shake flask. Then we pipette them on a filter and perfuse the filter, as you see here, with a medium. So constantly there's medium flowing through. But we can now switch the medium from 1 second to another by perfusing with a different syringe, and then remove, you can see my hands now, but remove this filter and immediately put it in a quenching solution. Okay, so this allows us, the setup allows us to change the condition for E. coli or for bacteria within a second. And that's what we're doing here. So we're interested in how fast can glycolysis switch. So when they're growing initially on pyruvate. So we have a carbon flux coming here. So there's gluconeogenesis, flux is going up. And then we suddenly switch to either glucose or fructose. So the experiment looks like here. We grow the cells on pyruvate in these shake flasks. They put them on the filter. And then within 30 seconds, so then we switch within a second to glucose. And for 30 seconds, we follow either on glucose or fructose. The dynamics of the metabolome. So now we remove in separate experiments these filters, measure the metabolome, and then we switch after 30 seconds back to pyruvate. So very short perturbation, 30 seconds from pyruvate to glucose and back to pyruvate. And the first question is, does E. coli actually know what's happening? Can it respond? Can glycolysis respond so fast? And for that, we use a special trick. We use isotopic tracers for our metabolomics experiments. So here's the pathway of glycolysis as you, well, the simplified version. And initially the cells were grown from here, from pyruvate. There was glycolytic flux going up. And now we add glucose. And suddenly the flux should be going down. The question is, how fast does that happen? And so by using uniformly labeled C13 labeled glucose, which easily in the mass spectrometer we can determine the fraction. Now we look at metabolites. So here's glucose 6-phosphate, and what you see on the y-axis is the fraction of C13, fully C13 labeled glucose 6-phosphate. And initially that fraction is 0, near 0, because it's natural abundance. And then within 5 seconds, already we are at 70 to 80% of all the glucose 6-phosphate up here in glycolysis is fully C13 labeled. So clearly very fast glucose goes in. And if we go to the end, phosphonol pyruvate, for example, here at the end, after the first sample, after 5 seconds, already half of the PEP molecules here are fully C13 labeled. So that means within 5 seconds, glycolysis has completely changed its flux direction. And the flux is already at the end. So it works very fast. Now to what we really want to do is, can we understand the metabolite concentrations that we measure? And so for the same experiment, it's the same logic, pyruvate, and then here's this glucose phase, and then pyruvate again. Now we're looking at the concentrations of, just focus please on the blue lines, on glucose 6-phosphate. There was a steady state level here. Then during the perturbation, very rapidly glucose 6-phosphate goes up. It almost reaches a sort of a steady state up here, much higher concentration when glucose is there. And as soon as we remove the glucose, it drops again. And then it needs a little bit of time, but eventually it will go back to a steady state here. Fructose 6-phosphate has a similar behavior. You can see that fructose bisphosphate has a somewhat different behavior. And the further down we go in glycolysis, the shapes look more and more strange. And then there's even, we're not going to talk about it, but the difference between glucose and fructose. Okay, so we can measure this at a reasonable accuracy, as you can see from the error bars. The question is, what does that mean? And that's very difficult to tell. And so here we need help from mathematics. And now we want to infer whether or not allosteric regulation is necessary to explain the measured concentrations that we saw. And the logic is illustrated here. So if you have a simple mathematical model where A in the simple pathway is providing a feed-forward activation to the second enzyme in this pathway, that would lead to certain expectations on the dynamics of metabolite concentrations. For example, A should go down very fast, but then C should really accumulate very rapidly because there's this feed-forward activation. If, however, we have a feedback inhibition from C on the first enzyme, we would expect a very different response. Now C would not be increasing very much, and we should be clearly able to distinguish this. And now all we do is we would take our measurements and compare it to the two possible scenarios. And in this case, it's obvious that measured concentration looks much more like, would be much more consistent with this particular regulation. Now, what I showed you was a mock example, and we need to be a bit more formal about it. And so we make a very simplified model of glycolysis. I don't need to go into the details. It has some reversible reactions. Some of them are irreversible, Michaelis-Menten kinetics, and we need parameters. So we take, so it's a fully reversible model. It can switch from pyruvate to glucose and back. We take the Km values from Brenda, and because we're not so sure about the in vitro data, we allow the values to vary tenfold higher or lower what is reported in the literature. We search in this entire space. And we fit the Vmax values of each of these enzymes from our pyruvate steady state data. So we have some indication of the parameters, but we don't pretend we know them exactly, so we leave a little bit of a larger range. So this is our model, no regulation whatsoever, just a simple model. And now we ask, is this model able to somehow capture the metabolite dynamics? And so here you see up here 8 glycolytic metabolites, and the dots are the actual measurements, and the line is the best fit even after days and days of running the search for a good fit of the model to the data. This is the best fit we can get. And here's 8 cases, so we not only look at the concentrations, but down here we also look at the dynamics of label distribution, so of flux, if you want. So the model is trying to explain the measured data as best as it can, given the structure that we give it, or that we define. And you can clearly see it doesn't work so well. There are cases when really the data doesn't look very good. Now, if we use an allosteric, if we add to this model allosteric regulation, so for example, if we allow PEP to inhibit phosphofructokinase and FBP to activate pyruvate kinase, which are known regulators, then the fit becomes much better for some cases, in particular for PEP and fructose bisphosphate. These are the very important regulators. So now this model would be able to explain the data much better, but not everything. You can see there are some other cases where it doesn't work perfect. So we wanted to, remember, we're in the business of generating hypotheses. We didn't want to know if the known regulation is enough to explain the data. We wanted to ask, can we find something new? And for that, we need to do our search a bit unbiased. And so we define 5 metabolites, which we allow to activate or inhibit any of the 9 enzymes that are in our mini model. And it's 5 metabolites because fructose 6-phosphate and glucose 6-phosphate always behave very similar, so they're co-correlated. So it's enough to look only at one, and the same is for fructose bisphosphate and dihydroxyacetone phosphate. So 5 metabolites, 9 enzymes, and this simple model that gives 90 putative interactions. And if we allow for pairwise combinations, we end up with somewhat over 3,000 models that we need to test. Okay, so now we have 3,600 hypotheses written down as specific models, and we are asking which of those can explain the measured data better. So how good are the predictions of these models? And so we include this regulation on top of the simple unregulated model as a power law term for those who are interested in it, by which now there's the models need to fit an inhibitory or an activating regulation event in the model. And so now with 3,000 something models, we can take our finger to say, oh, this model is better than another. So it's a formal criteria. It's called Ikaike information criterion. And it measures how well a model can describe the data, but it gives a penalty if the model has more regulation events, because if you have more equations, it's easier to fit the data. So that's what you see here. And basically, we have here 90 interactions in combination with 90 other interactions. And red means this particular model with regulation scores less well than the unregulated model. It's only the green models that are better than our unregulated model. So you can see most of the regulatory interactions actually play no role, but there are some that seem to play a role. At least there's some green, for example, for this particular case here. So that looks, and that's exactly the information that we use. So first we ask, how often does this interaction here, this is, I can't really read it right now, but it's a particular inhibition of phosphofructokinase by PET probably. And it occurs in just about every other combination with other regulation events. So we ask, how often does one particular interaction occur? And the second criterion is, how good is the best model with that? So that is, how green is it? So here, for example, you see a few very bright green spots. That's probably more important than, at least it's a higher probability than this very light green up here. So if we use this criterion, most of these all of these red models would be down here below 0 because they're worse than the model without regulation. Here at the point 0 is the model without regulation, and everything that is in this quadrant here is now better than the model without regulation. And there are some regulatory inhibitions that, some regulatory interactions that really seem to be very important, and they are the ones up here in this upper part. And the black ones were actually known ones. I'm going to show you one in a second. And the white circles are those that are really have never been reported. For example, pyruvate being an activator of fructose bisphosphatase. Right now, only a hypothesis. So let's see how good our data is if we take a known case. And so a known case that in the literature was known is that fructose bisphosphate is activating pyruvate kinase. So that's a classical feed-forward loop, and the top part of glycolysis is informing the lower part of glycolysis that it should be more active. And that has been very well biochemically characterized. And we find it, so this is, if you wish, a positive control. And now we want to know how good is our model to explain basically the data. And so here is if we simulate this experiment from pyruvate, switch to glucose and then to pyruvate, the black line is what we would predict was a native pyruvate kinase that has this feedback, this feed-forward activation. And because an enzyme exists, or a mutant enzyme existed that does not have this activation of FPP anymore, we can simulate what would happen before we even do the experiment. Now we would expect that fructose, in such an insensitive pyruvate kinase mutant, fructose bisphosphate concentration should, in the shift, continue to increase, and then take much longer to come back to steady state. And that is because the lower part of glycolysis doesn't yet know that it should be more active, and therefore glucose accumulates a little bit as a fructose bisphosphatase over time, as fructose bisphosphate. And if we do the experiment, we find qualitatively exactly that. So then afterwards, we got this mutant here, we did the experiments, and indeed fructose phosphate concentrations go up much, much higher. So the models are actually able to capture the dynamics fairly well. And then the last thing that we want to see is, can we now find something new, novel regulation that isn't known? And so I pointed already to the pyruvate activation of fructose bisphosphatase. So this is now the opposite effect. That's a molecule from lower glycolysis that informs the upper part of glycolysis that it should be more active. And so this is one of the very strong predictions that we made, or very high confidence on this in this particular prediction. So the way to test this, then we purified fructose bisphosphatase and measured its relative activity. And the relative activity without pyruvate is 100. And now if we add more and more pyruvate, you can see that the activity of this enzyme goes up quite a bit, 3- or 4-fold. So yes, there's an activating effect of pyruvate on the activity of fructose bisphosphatase, which was basically what this interaction predicted here. Okay, so this is the end of this particular part, and I wanted to mainly illustrate to you that dynamic data are really extremely useful, but we need a good mathematical formalism to be able to interpret it. But the experiments that I showed you were fairly tedious. To do these second measurements, it takes quite a bit of time. And so just as a teaser at the end, we developed a technique that we call high throughput metabolomics, because it is really fast. We can measure many samples per day. And the trick is we remove all upstream chromatography and shoot our samples directly into the mass spec. And so that allows us, without chromatography, on measurement is 1 minute, so we can measure 2,000 samples a day. It's semi-quantitative because we don't have any good calibration. We just know if something is increasing or decreasing, but we don't know anything about the absolute concentrations. In a typical experiment, there's somewhere in the range of 500 to 600 metabolites that we can measure. There are many different parts of metabolism, and you can probably imagine that there are many shortcomings of this method. One of them is because we have only mass to actually annotate a peak. Things with the same mass cannot be distinguished. So glucose 6-phosphate, fructose 6-phosphate will be the same. And the matrix must be constant. We can only compare things with a highly, highly similar matrix, because there's many matrix effects. Without chromatography, we have matrix effects, but we have to ensure that it's the same effect in all of the samples, which can be done and actually works fairly robustly. And so this was, we published this method already 2 years, or maybe 3 by now ago. Hannes and Toby did that. And we went in fact, the method was published already a bit earlier, but what we wanted to do is we wanted to exploit it to do dynamic experiments. And so I'm showing it to you here in the simplified graph. So we cultivate our cells next to a mass spectrometer. So you don't see it here in the picture, but up here would be a mass spec, the QTOF. And now this culture, there's a loop. We continuously through a pump about 20 microliters of cells in a continuous circle. And then we can computer control the valve and change it such that 2 microliters of medium plus cells are injected directly into the mass spec. So the real setup is here. Here's the cultivation of cells. Here are these valves, and here's the pump. And so this tube here goes to the mass spectrometer right here. So now we shoot 2 microliters of cells plus medium, must be low salt, otherwise the mass spec is not going to say thank you. So it must be low salt, but we can do this for many cells and shoot 2 microliters into the cell. And I just want to give you one example, not a real story, just one example so that you see how this looks. And this is work done by Kartik Sekar. But we are interested in how bacteria deal with sporadically available nutrients, which really is a reality for cells. They don't have a continuous supply of something. So what happens when they get a little shot of something? And so we modulate its glucose that we're giving to the cells, but we give them only very rare shots of glucose. Sometimes we give them much more frequently, and we can change the frequency of these pulses. And so the experiments, just so that you know how this works, we grow the cells in OD of 1, we resuspend and diluted M9 medium without glucose, and we keep them in this for 2 hours. So we starve them, and then we do these pulses of glucose. And what do the cells do with the glucose? That's the question, the initial question that we have. Now we have very different questions, but initially we were just wondering, where does the glucose go? And I'm showing you here 4 metabolites, and only the pulse here. This is a very low feeding rate. So they get a very short pulse of glucose at 5 minutes, and then the next one at 35 minutes. So half an hour starvation in between. And when they get this tiny amount of glucose, which is not enough to grow, they cannot grow. You can see hexose phosphates. You see, this is now these real-time data. You can hardly see the points. There's 2 or 3 points when there's a peak really going up quickly, and then it goes very rapidly back to steady state. Next peak the same, shortly up, maybe we can measure like 6 points per minute, so every 10 seconds, so maybe 30 seconds is a pulse. If you go further down in central metabolism to glutamate, it's now somewhere close to the TCA cycle. The peaks are a little bit wider. It takes longer, a little bit longer for the carbon to go there after the pulse, and it also takes longer to go back to steady state, but it is not so different from up here. So glutamate also in central metabolism, the glucose rushes through here, goes to glutamate, and then is also at 0. But we see accumulation of amino acids, for example, in the pulse, in between the pulse periods. And this is a compound hypoxanthine that's related to DNA and RNA elongation. So biosynthetic building blocks are actually already synthesized, even though there's very little glucose that we're feeding. Okay, so what is really happening, and so I can show you, if you pulse much more, double as frequent, nothing changes in the upper part, has very short peaks, but still there's an accumulation of phenylalanine, hypoxanthine, and many other amino acids, for example. So it really looks like carbon is rushing through central metabolism in just about no time, and then slowly accumulates between the pulses in building blocks for cells. And with that, I'm at the end of my talk. So the conclusion is that dynamic metabolome data delineate confounding from indirect consequence. Dynamic metabolome data delineate confounding indirect consequences from the direct consequences. Mechanistic models can enable hypothesis generation. In our case, they were really key to assess in vivo relevance in a larger network context. And allosteric regulation directly controls the flux magnitude in short-term dynamics, and currently we're working a bit more on longer-term things. And if much of this was over your head or you were not so interested, the real take-home message is from the first part that there's really simplicity in cellular regulation, at least in microbes. It's not so terribly complicated if we just know exactly what's happening. So the point was that we have only 3 regulators that explain 90% of the gene expression regulation. Hypothesis generation for metabolomics data requires absolutely computational methods. Without it, we are almost completely lost, or you must have a very specific hypothesis. And dynamic data, please keep this in mind, is much more informative, in particular for metabolomics. This is even more important than for other omics because of the much less confounding indirect effects. And I think I mentioned the people along the way that did the work. And with that, I thank you for your attention, and I'm happy to take questions, and we'll see how this works over the internet. Thank you.
Thank you very much. I would like to thank you, Professor Sauer, for this great and insightful presentation. If there are no remaining comments of the presentation, I would like to proceed to our question and answer session. During the webinar, you have submitted some questions, and I will request our speaker to address as many as possible. If you still have a question in mind, please make use of your question tab in the webinar app, and we can address them in the meantime. So well, I would like to therefore start with the questions. We have so far 2 questions. The first one is related to the first part of the talk, and it says, compared to simplistic prokaryotic metabolic blocks, how such models are feasible in complex organisms like primates or, say, non-model organisms, especially when diet, environment, multiple organelles, and the gut metabolism interplay affects a lot?
So, I mean, in the first part, we didn't really have flux. So I'm not exactly sure what the question really is trying to ask. You said it was about the first part, right?
Yeah, yes, specifically global and specific regulation of the transcription factors.
Okay, for the global and specific regulation. So that part is definitely the same. That has nothing to do yet with flux. It's just gene expression, also in mammalian cells, changes a lot based on how fast cells are dividing or not dividing. And so if you have cancer cells, you will see many changes in gene expression simply because these cells are dividing faster. And the same is actually true for the proteome. there are changes that have nothing to do with specific regulation. They are just a consequence of the physiological state in which the cells are and how fast they're growing. So we call these, it's not specific regulation, it's the influence of some global factors. Is that what the question was going for? I was confused because there was no flux in the first part.
Basically, so far I understand as well is
okay.
There's going to be an effect regarding the diet, for example, depending on a more complex organism as well, whether the environment will affect this. But please, the person who made this question, feel free to make another comment and possibly specify a bit more, and we can certainly address this in a better way.
Okay.
But in the meantime, we can pass to the next question, and this is related to the second part of the talk regarding the allosteric models. So, in addition to the allosteric models explaining the data, wouldn't not benefit from the allosteric models explaining the data wouldn't not benefit from evaluating their corresponding protein levels using, say, proteomic approaches to see the protein?
So the answer is no. And that has to do with the starting point. You can still see my screen, I assume. So we are looking at very short responses. And so for here, it would help a little bit if we knew the absolute concentration of an enzyme, but because we have the Vmax value that's just so that everyone is on the same page. A Vmax value means now, if we had the concentration, it would help us a little bit to parameterize the model in the beginning, but Vmax is a good approximation because it is a factor of the specific activity and the amount of enzyme. And so we already estimate from our flux data the overall in vivo flux through the maximum flux through this reaction. So it wouldn't otherwise not help, because in the 30 seconds where we look at changes, the metabolites are changing big time, but the proteins would remain completely constant. In 30 seconds, there are no protein level changes. Maybe post-translational modification, that is a possibility. That could also still happen at that time scale. But concentrations wouldn't matter.
Okay, thank you very much. Apparently, we don't have any other questions, but please feel free if you have any questions to reach out the Metabolomics Society or directly Professor Uwe in case you are interested. Well, with this, I would like to, if there's any other comments, I would like to finish this webinar edition. And I would like to invite you to our next webinar series that will be in March. For that, I'm just gonna show. Okay, we have a last question. Okay, no, it's basically explaining the first question, but I think you addressed it. Flux is not related in the first question. Yeah. Yes. Finally, I would like to thank you one more time, Professor Sauer, and all of you who joined the session. We're constantly working in the committee and this network to bring initiatives for training the next generation of scientists in metabolomics and all related fields. But therefore, any feedback or suggestion about this network is always welcome and very much appreciated. I would like to thank you again for this attendance. If there's no any other comment, have a good day regardless of your location worldwide.
Okay. Thank you very much.
All right. Thank you very much.
Thanks for organizing.
Thank you very much.
Bye-bye, everyone.